The skin is the largest organ in the body. And not only is it a barrier, but it has a very active immune system. When we think of inflammatory skin disorders, psoriasis, when we think of atopic dermatitis, they all have a component of skin barrier disruption. And so many of the diseases in dermatology become a vicious cycle between skin barrier disruption and inflammation. Patients also experience things like itch, burning, stinging, and it affects their quality of life. And my job as the scientist is understanding how to break this loop. In the big picture, my laboratory is doing what is called structural biology. We are trying to understand the structure of how proteins, nucleic acids, and other macromolecules work in the skin, and how drugs that target different skin diseases work. And then in certain genetic diseases where those proteins are in error, we want to understand what has gone wrong and how can we fix it. There's no one else in dermatology that is running a primary NIH-funded research lab that's dedicated to structural biology and answering those difficult biochemistry questions through the approaches of X-ray crystallography, cryo-electron microscopy, and if needed, nuclear magnetic resonance imaging. So X-ray crystallography is a particular technique that allows you to understand the atomic level structure of proteins, nucleic acids, and their complexes. What you have to do in crystallography is take a very pure protein, for example, and put it in the right solution that allows it to precipitate. And when it does it just right, it forms a crystal. And that crystal is a perfectly aligned lattice of lots and lots of molecules of that protein of interest. And then when you hit that crystal with X-rays, that crystal then diffracts the light. And then what we can do is use mathematical approaches to take that diffraction pattern and turn it into actually a three-dimensional model of that protein. And that allows us to determine the structure at that atom and bond level. And then we can take that information and ask those critical questions. What does this tell us about helping the human patient? One of the wonderful things about being a physician scientist is I get to see what it's like in the research lab to make a discovery, but I also get to talk to patients face to face. And I really get an understanding of what matters to them. And the advantage of this is it allows me to then come back from a translational perspective and understand what is going to help deliver better patient care. This is also why I really am excited to be a part of clinical trials here in dermatology at Yale. I love helping patients. I love it when patients feel better and are happier with themselves. In the end, all of what we do, whether it's the research mission, the clinical mission, or even a clinical trial mission, it's all about advancing patient care, developing new therapies for rare genetic skin diseases for which patients have nothing yet.